Hello everybody, I'm Phil Gribbon, President of the SLAS Board of Directors for 2025, and I'm really proud to be part of this diverse and talented community, and I'm planning to use my President's messages to have conversations with a variety of SLAS members. Please enjoy this first chat with Nicola Richmond, who's the Chief Scientist for AI at Recursion in London, and also will be the co-chair of this year's SLAS Europe Conference and Exhibition in Hamburg. Nicola, it's great to catch up again, and as you know, some folk will get a chance to meet you in a few weeks in Hamburg at the SLAS Europe meeting, which is kicking off on the 20th of May. But for those who aren't lucky enough to be there or don't know you too well, could you give me a little bit of introduction to your career and your journey and what's brought you to the Recursion?
Yeah, okay. So, yeah, I've been around for quite a long time now. So I identify as a mathematician. That's what I studied at university, and I was super theoretical. And then I hate to admit it, but by accident, I fell into the pharma industry via a postdoc in cheminformatics. And I started my, let's say, pharmaceutical journey at GlaxoSmithKline and spent an 18-year period working at GSK. And I realized that to some might sound a bit dull, but I had lots of different jobs while I was there. So I spent time in cheminformatics, computational chemistry department, and there I worked very closely with high throughput screening groups and got very interested in lab automation. And I also was connected to SLAS through my GSK career as well. I spent time in a general data science group and focusing on bringing really state-of-the-art analytics methods into GSK. And at the time, we weren't doing much in the neural network space. And then we started getting interested in building convolutional neural nets for looking at and understanding cell painting data, which then connects us to recursion in a way. I spent time thinking about developing methodologies to accelerate antibody-based drug discovery. So I've worked in both small molecule and therapeutic antibody-based drug discovery. And then the last 4 years of my role at GSK, I was in the AI and machine learning department, and there we're very focused on using AI in conjunction with human genetics and functional genomics to identify really novel targets with genetic validation. And then I parted company with GSK about 3 years ago, and I joined Benevolent AI, leading their AI function. And there the focus was very much on target discovery, but using natural language as a data type. And then 6 months ago, I joined Recursion via the combination with Exscientia. And yeah, here I am today.
Fantastic. It sounds like a really exciting journey. I think I'd probably share some of those steps with you, but not all. But maybe it'd be really interesting to sort of jump into some of the detail and especially around how within organizations like Recursion, you're able to successfully orchestrate all of those different elements of the design, make, test, analyze process and really create an efficient drug discovery workflow. Would you be able to share some of those details with us?
Sure, yeah, I'd be really happy to do so. I've invested in building a fully integrated design, make, test, analyze platform, and that starts with the design aspect. And there we need a design cycle strategy. So, for example, it could be, oh, yeah, we really need to improve solubility. And then we also need a seed molecule. And in a lead optimization campaign, that's likely to be the best molecule that you have to date. And then we have AI-based molecular generators that will build populations of molecules for us with the aim of improving solubility in this example. But what's important is that those molecules that are generated by our AI approaches are actually synthetically tractable. So once we've done the design aspect, and we're able to score the molecules as well. So we have a suite of property prediction models that will give us predictions, and in our example it's solubility. So we'll have a solubility prediction, and then the next step is that we'll choose a subset of these molecules to make in our facility at Milton Park. And once we've landed on the set that we want to make, the next step is to work out, well, how do we actually make these molecules? And we want to be able to make these molecules in such a way that we get access to them as quickly and as cheaply as possible, and we can make the highest quality molecule with the greatest yield. That's what we're aiming to do. And we have models in our platform that will do retrosynthesis prediction and also reaction outcome and conditions prediction as well. So this really allows us to make the molecules in the most efficient way. And then once we've made our molecules, and that's done with our automated synthesis platform, and this is fully automated. So a quick anecdote. So the first time I visited the facility at Milton Park, I was blown away. I mean, it really is super cool. And I got to press the make me paracetamol button on the laptop, and then literally an hour later, paracetamol popped out of the instruments at Milton Park. And then co-located with the automated chemical synthesis platform is rapid biology automation platform, and that's where obviously the testing takes place. And what's cool is once we've got the results, they're automatically fed back into our system, and those data are picked up by our ML platform, and that then triggers automated retraining of our models. So our models are continually kept up to date, and also the human computational chemists, because we do have humans in the loop, they're also able to look at the data in light of the design cycle strategy and see how the cycle has performed, and then that really informs the next design cycle. And we just keep going and going until we get from our leads to our candidate molecule. And actually I've got a really nice example of which is our CDK7 inhibitor that's currently undergoing dose escalation studies, and there we went from hit to candidate in 11 months, synthesizing just 136 novel compounds. So you can really see the platform working in action, actually having an impact on programs, which is really cool.
That sounds really incredible, and I think from the outside, I'm working with the logo in the corner there for a public organization, and much of what we do is are generating data sets against various targets, and many of them get deposited on public resources like Chembl or the European Chemical Biology Database. And I imagine that there's some utility in using those data sets and those data resources. But I was sort of interested in sort of understanding to what extent they're useful to you, and what you see as the sort of what works well with the using public data, and maybe what's not quite so good and where you've seen some issues with that.
Yeah, so when we're in the business of building models, access to high quality, diverse data is really important to our industry. It's an industry that's 100% data driven. And if we think about some of the successes in AI outside of our industry, for example, we see these big models that are able to generate convincing text and really convincing looking images. And one of the reasons that they're so good is because we've got a ton of text data out there and digital images out there in the public domain. I guess the other thing that we shouldn't forget, though, is that it's not just about the data, but we also need access to massive compute, and that's advanced at an astonishing rate as well. So we've got the data, we've got the compute, and we're now starting to see AI working tremendously well in these fields. And then in our domain, at Recursion, we've invested heavily in data generation, particularly in the phenotypic space, so in cell painting space, bright field, so microscopy. And actually, we recognize the importance of making data publicly available, and we've actually done that. So we've placed around 4 million cell painting images into the public domain today, and these images are primarily of HUVEC cells, and we've perturbed them either with a gene knockout or a compound treatment. And alongside those data that anyone can download and build models with, we've also released a really large what's called a vision transformer model called Open Phenome, and that's been trained on cell painting images. And if people are interested, they can access that model on Google Cloud's Vertex AI model garden or through Hugging Face. And the data sets are on rxrx.ai if people want to take a look. And the reason that we think it's really important to put data into the public domain is that it helps advance our industry, and it's analogous in a sense to what I described earlier with the literature and images. They're all available for the community to use. And we want to advance our industry through the AI research community that, like us, is really trying to change the way we discover and develop medicines, because it's really hard. And then in terms of what works really well and what's useful and what's not useful in data sets, I'd say that it really depends on the problem that you're trying to solve, what your modeling aims are, and how you want to use your models in production. So, for example, if your aim is to understand disease biology, biology is big and complex, and we need lots of diverse data in order to help us understand biology. And that's why at Recursion we've been generating over the decades now petabytes of data, and the data that we've released into the public domain forms about 1% of what we have in-house. And having access to massive data sets really allows us to leverage some of these super powerful neural network based models. And we use these models to build what we call our maps of biology. And we use these maps to both initiate programs, so that might mean in traditional speak, target discovery, but we also use our maps to find chemical start points. And then this is quite different to the scenario that I described earlier when we're in, say, a lead optimization campaign or a hit to lead campaign. Here we can get away with using more traditional machine learning approaches and physics-based approaches as well, because there we're really interested in looking at localized areas of chemical space, and we're often in this low data setting. And the emphasis is still very much on having high quality data and also having reliable negative labels, because bench scientists really like to generate data and in a positive sense. So what does that mean? If you think about the pharma funnel, we're always progressing things that we like and we're interested in, but machines need to know about what's bad as well as what's good, and they have to have some exposure to both the good and the bad during their training process.
I mean, it's very exciting to hear about the release of data into the public domain, and that actually is very complementary to some of the work we're doing in EU Open Screen. We're just in the process of cell profiling over 100,000 compounds in our library, and those data, I think by the end of this year, will be made available for reuse by the wider community. I guess we talked about public data and also data coming from your sort of in-house testing processes. But when you're working with third parties and collaborators, and they may have a large amount of pre-existing data, are you able to sort of successfully integrate that into your workflows, or do you find some challenges in the harmonization and integration of those data to try and sort of drive the model development?
Yeah, so we do leverage partner data as well, and we have fantastic partnerships with the likes of Tempus, Helix, and then most recently Health Verity, and this gives us access to millions of patient data records. And I talked a little bit about our maps of biology. I think it's probably worth saying what they are, and at a super high level, what our maps are. We've got these HUVEC cells, so that's our cell line, our workhorse cell line, and we've applied gene knockouts to those cells, and also we've treated the cells with compound, and what we're looking for are gene knockouts that give us a significant change in phenotype, but then we're also looking for compounds that give us a similar change in phenotype, and this allows us to link genes to compounds. But what we need to also be able to do is link genes to disease, and we do that through our partnerships. these providers with these really invaluable links to disease and, of course, to the patient. And then other areas that I think might be worth spelling out is in our maps of biology. So I mentioned that we're looking for interesting genes and then interesting or good relationships between genes and compounds that give us similar phenotypes when we compare knockout with compound treatment. Sometimes we can't find chemical matter for genes that look really interesting, so we tend to refer to this as the dark space in our maps. And here we've worked really closely with Inamine, so we've been using our machine learning approaches to build 10 compound libraries against 100 key and clinically relevant genes, working very closely with Inamine, which is really exciting. So that allows us to shine a light on the dark spaces in our maps of biology.
That sounds very good. And I have to say, I got a chance to see your talk in San Diego, where you also talked about some of the sort of kinetic dimension in cell painting that you guys had started to introduce within Recursion, and to sort of doing digital phase or phase contrast type imaging. And I think that was really, really exciting, because I think that's often overlooked element of the phenotypic response is not just the cell line or the concentration, but it's the time that you choose to take the measurement. So yeah, I've encouraged my team to try and be a fast follower there, to see if we can also implement some of those methods. But no, that sounds really exciting.
Yeah, so yeah, very true. You know, so I've talked predominantly about cell painting, but actually for that example, we're leveraging bright field, and obviously the advantage with bright field is that the cells, you don't have to kill the cells, so you can test them at multiple time points and see how they're doing, and also you can test the plates with other phenotypic endpoints as well, which is really cool. Very excited about that avenue.
Yeah. And I think another sort of very exciting and interesting development just in the last few weeks has been this announcement from the FDA around their plans to sort of phase out animal testing requirements, especially in regards to the development of monoclonal antibodies, but potentially other types of drugs as well. And AI, as well as lab on a chip technologies, are positioned as being able to sort of provide appropriate new approaches and new methodologies to substitute for those animal models, the use of animal models. So I'd be really interested to see how you view that development, and what the implications are for organizations like Recursion when they're developing their own compounds and also potentially working with partners.
Yeah, so for me personally, this is really music to my ears because it aligns so well with the 3Rs framework, which is the aim to reduce, replace, and refine animal experiments. And this framework's been around since I think it was 1949 it first came out. So it's great to see the FDA taking this line. And then in terms of how this relates to us at Recursion, so Recursion is over a decade old now, and we've spent our entire lifetime focusing on leveraging human cell-based screening and AI-driven patient-centric models to really help us understand disease biology through our maps that I was talking about and our partner data. And then also we have our compound property prediction models as well, which allows us to prosecute the chemistry phases of drug discovery in an efficient way. So I feel like we're really well placed to respond to this FDA plan. And just as an example of some of our modeling capabilities, so we have this really quite, in property prediction world, quite a large model. It's 3 billion parameters. It's called mold GPS, and that's for ADMET prediction. And just to your point, Phil, about the importance of having different data that are harmonized, here we're leveraging a public multimodal data set of around 5 million molecules, and those molecules have been subjected to transcriptomics, bioassay, DFT simulations, and we're taking these multimodal data, and we're pre-training our model so it understands all these different aspects of biology, as well as physics with the DFT, and then we're fine-tuning our model for ADMET prediction, which again is like super important, and it's achieved state-of-the-art performance when we compare it to benchmarking tasks that are out there in the public domain, which is really exciting.
I'm going to put you on the spot now. If you only had 3 sets of data and sets of parameters that you were had access to and you were using as part of your optimization process in a hit to lead or a lead to candidate program, which 3 of those would they be? What's the priority? What's the minimum viable product, as it were? How does it look?
Oh, okay. So I think first of all, we need to understand potency, and for that we need potency data, and ideally in a translational system. And what does that mean at Recursion? That means that we establish potency either through phenotypic or target-centric means. And then next, if I had to choose, it's like, well, we want to know that our molecules are going to have a good PK profile and they're going to be safe. So I'd really like some ADMET data. I don't know if that counts as one data source. And then finally, something that I've talked about earlier was it's really helpful for us to have access to CRO logistics data, because that really helps us understand which building blocks are available, how much they cost, how long it would take to get them. And when we're thinking about all those different synthetic routes that I talked about, this helps us pick one that gives us confidence that we're going to be able to make the molecules in the shortest possible time frame at the cheapest cost. So that they would be my 3.
Sounds great. I think there's hope for the rest of us to try and follow you, because we don't have access to all of the data and resources that you have. But that's a very good point there for people sort of starting off in the field. Nicola, it's been a really very, very interesting discussion. I'll be looking forward to welcoming you and showing you our Hamburg and all the nice, great science that's going to happen, but also the great city that it is as well. So looking forward to catching up with you then, and have a safe trip over from the UK. Thank you.
Thanks, Phil, and I'm really looking forward to being part of SLAS Europe.